Okay. Hello everyone, my name is Xinyu. I come from Wesleyan University as a PhD student. Today the topic I want to share is distinctive signatures of pathogenic and antibiotic resistant potentials in the Hadal microbiome. The trench is a unique ecosystem that have been deeply explored on Earth, with a depth around 6,000 to 11,000 meters, and exists in zone by narrow topography V-shaped depressions. The environmental conditions of Hadal is very special, with high pressures, low temperature, low dissolved oxygens, and low nutrient concentrations, and so on. Predominant factors in the Hadal environment, including protobacteria, chloroflexi, some archaea. One of the biggest threats to our ocean is man-made pollution. At more than 10 kilometers depth, the Mariana Trench might seem remote enough, but plastic waste pulps and mercury were found there. The PCB bioaccumulate in the Mariana was 50 times more contaminated than crops from paddy fields fed by the Liaobo River, which is the most polluted river in China. This data indicated that serious man-made pollution occurred in the 2006 year, and it will spread all over the world with the human production activities and the global water cycle. A lot of research describe its distribution and spreading mechanisms in the environment. The ocean is considered as a global reservoir of ARG. A frequency of a horizontal gene transferred in the control group is only -7 power of 10, but ocean is about -4 powered to -2 powered of 10. So the ocean is the place where HGT highly frequency occurred. Researchers are actively studying in microbial samples from the Hadal zone. However, the pathogenic and antibiotic resistant potentials of the microbial populations therein have never been closely analyzed, especially man-made contaminants, including POPs, antibiotics, may also signify yet to be discovered as a pathogenic disturbance on the local microbiota. In order to explore the composition of the microbial communities, the virulence genes and ARGs distributions, and its potential mobility at the deepest point on the Earth, discuss the difference of ARG and MGEs between the seawater and sediments. We target 4 Mariana Trench sediments and 1 oceanic basin sediments and 3 seawater samples to analyze. They came from our self-sampling project with the help of Professor Cui and Dr. Liu and some open access dataset. So based on qPCR result, we found that the microbial community seems to be more prosperous in the bottom of the challenge deeps than in the step wall and the basin sediments. It possibly because the V-shaped depressions of the trench allowed more accumulation of rich organic pumps from various sources such as dead animals and plants. Intriguingly, 12 and 9 virus phyla are revealed from the challenge deeps, but in the basin sediment only contains 3 virus phyla. The result indicated that, compared to the shallow locations, viruses are more diverse in the Hadal zone. Homolyzing and repeat toxin encoding genes were shared in all samples, but some special virulence genes, including botulinum neurotoxins, tetanus neurotoxins, and Clostridium glucosylating toxins, were found only at the bottom sediments, and we did not observe any related genes in the other samples microbiota. This result indicated that potential selective enrichment of these special toxin genes at the challenge deep bottom. The abundance and richness of ARG greatly different across the sediment seeds and inhibit potential distinct decay law. There is a clear composition distinction of ARG profile between the water microbiota and sediment microbiota. Multi-drug, MLS, and beta-lactams form the resistant group in the sediment as the predominant parts. But in the seawater, multi-drug is the most important ARG. and multi-drug efflux genes ACRB, MEXT, and sulfonamide resistant genes SU1 was strongly selective enrichment at the seawater samples. That may be the evidence of the Hadal microbiota adaptation to the inhibitory chemicals of the pathogenic origins. We recovered 127 to 187 type ARGs high abundance from seawater samples. In other words, the Hadal seawater contains an extremely high levels of ARG that is comparable to the one reported in the pathogenic impact wastewater and sludge, and exceed that of normal drinking water, river water, soil, and the ocean sediments 2 orders of magnitude. The mobilome of microbiome enable bacteria to quickly acquire the novel traits to survive environmental changes through HGT. So we predict the mobile genetic elements. Overall, the Hadal sediments and water show clearly different richness and composition of MGEs. However, both habitats mobilomes were dominated by integrated genes, especially those encoding by the phage, implying the importance of the phage-mediated HGT mechanisms for evolution of Hadal microbiome. Mariana virus are the main drivers of the evolution of both host and virus assemblage in the ocean environment. Conjugation and plasmid-related MGEs account for a negligible relative percentages in the sediments, whereas they were found in the seawater. Revealing plasmid and conjugation-mediated HGT should be much more common in the Hadal seawater than the sediments. That may be due to the sediment's microorganisms' lack of sufficient physical contacts, essential for conjugation. With the low biomass environment, the flowing seawater provides the opportunity for the physical contacts of the microbiome. So compared to the basin and the step well, the challenge deep bottom harbor 7.05 and 4.67 fold of the mobilized ARG, and mobilized VGS were also more abundant in the bottom. Compare the basin and the step well, the challenge deep bottom have about 59 mobilized VGS failed to be classified. This result reveals that the extreme environment of the change bottom may enhance the propagation of the mobilized ARG and VGS, which originate with frequent HGT events and would reflect long-term adaptive traits of the microorganisms inhabiting the bottom sediments of the challenge deep. This study reported the landscape of the virulence factors, antibiotic resistance, and mobilomes in the sediment and seawater microbiota, receding Hadal environment of the deepest ocean bottom on the Earth. Our work unraveled constructing the unique features and virulence genes ARGs and MGEs in the Mariana Trench bottom, providing new insight into the eco-environmental and biological process underlying microbial pathogenic resistance and adaptive evolution in the Hadal environment. So very thanks to my supervisor, Dr. Zhu, and all the team members from our lab, and thanks to the sampling help from the Professor Weicheng Cui and the virulence gene analysis by the Dr. Tao and his students Liu Qinghe. That is the brief introduction of EMB lab. So if you are interested in our work, you can contact us or follow our WeChat and website. Thanks for your attention.